So there was a terrorist activity which happened and led to the destruction of the World Trade Center. In the aftermath of the 9/11, the US Postal Department received various mails or envelopes in which there were white powders. Those powders were nothing but spores of Bacillus species, and there was a scare of anthrax. Now, these spores can enter the human body and cause disease. Normally, till now, we have discussed most of the organisms are potentially virulent organisms in their vegetative form. But some of the spore forming organisms, if their spore enters the human body, can cause human disease. I think you have heard about a disease called tetanus. It is caused by Clostridium tetani. So, where does this Clostridium tetani? So, one more important, Clostridium is an anaerobic organism, right? So, it cannot survive in the surface because the surface is exposed to oxygen. Yet, human beings do get the infection. How? From the spores of the Clostridium bacilli. The bacilli remains in the subsoil in the vegetative form. Subsoil is anaerobic. Okay. When it comes into the topsoil or the surface, it comes in contact with oxygen. So an anaerobic bacilli would not be able to survive. So what it does, it converts itself into spore. Now this spore is commonly found on the surface of the soil. And now what happens? Where suppose a person gets into a road traffic accident and injures the limbs. The limb will have open wounds contaminated with soil, and this soil will contain the spores of Clostridium bacilli. If that wound is not properly cleaned, what will happen? This spore now will get a very conducive environment on the human body, and the spores will germinate to form Clostridium tetani bacilli. And this bacilli will start producing toxins. And when this toxin is absorbed, the person develops the disease that is called as tetanus. So, long story short, that is the importance of the organisms which can form spore. So, not only the vegetative forms are infective, but also spores can also be infective. Okay. Other than these four, there are many other organisms which are gram-positive bacilli can cause human disease like Listeria, Erysipelathrix, and also Actinomyces or Nocardia. Now, I think you can see all this. So, gram-positive rods, now we can divide them whether they are forming spores or non-spore-forming. Among the spores forming, we have Bacillus species and Clostridium species. Among the non-spore formings, then we look at the morphology or the shape of the bacilli. It could be regular shape or it could be irregular shape. Irregular means, what do you mean by irregular shape? They will have various pattern in their morphology. Till now, whatever bacilli you have encountered, especially the Enterobacteriaceae, they have regular shape. When that shape is not regular, we call them as irregular shape. So, among the regular shape bacilli, we have two types of organisms. One is called as Listeria, another is Erysipelathrix. Listeria causes Listeria meningitis and neonatal infections. We will talk it about later on. Among the irregular bacilli, which have irregular shape or different staining properties, we have non-acid fast, acid fast, and filamentous. Now, acid fast, I have already told you, it belongs to the group of mycobacterium. Among the non-acid fast, we have Corynebacterium and Propionibacterium. Corynebacterium causes Corynebacterium diphtheria causes diphtheria. There are many other species of Corynebacteria which are also part of the commensal. And finally, we have filamentous gram-positive bacilli among which we have actinomyces and nocardia. Actinomyces causes a disease called actinomycetes. The name suggests mycetes like a fungal infection, but it is not a fungus, it's a bacilli. The bacilli, the shape of the bacilli is so long that under microscope it might resemble a filamentous fungi, but it is not a fungi. Similarly, another filamentous bacteria is nocardia, which is also acid fast. It causes nocardiosis. So this would be the classification. Now, why this classification is important? That is how we proceed for the diagnosis. Now, first let us look at bacillus. Now, this bacilla will form spore. I think you can see on this picture. This is a vegetative cell and this is a spore. When the sporulation is complete, the spore is liberated from the bacilli itself. And over there is a ground stem. Now, the shape, if you can appreciate, they are very long chains and each bacterium is shaped like a box shape, almost a rectangular shape. But because they are aerobic bacilli, they are catalyst positive, and among the diseases which they cause, the most important disease is anthrax, caused by Bacillus anthracis. Another important disease is caused by Bacillus cereus, which causes gastroenteritis, or commonly encountered with food poisoning. Among the other organisms, which include Bacillus mycotis, thuringiensis, but the infections are not very common. Can you name any other use of this bacillus? They are also used as a control in sterilization process. Geobacillus stereothermophilus. These are also used in the sterilization control. Okay. Now let us look at Bacillus anthracis. Can you appreciate very long chain and single individual bacteria looks like a rectangular rod shape. Now I think you have already been taught in anthrax in your class. So what is an anthrax? It's an invasive disease caused by Bacillus anthracis. There are many types of presentation. It could be a pulmonary presentation, it could be a gastrointestinal presentation, or it could be a cutaneous presentation. Commonly, this disease is of a veterinary origin. The cutaneous transmission is most commonly seen in people who are handling animals. So when they carry the animals on the back or shoulders, they commonly get infected by in the form, and the form commonly seen is cutaneous anthrax. I think you can see the cutaneous also over there. This is the cutaneous form. The most dangerous form is the pulmonary form of anthrax, which is acquired when a person inhales the spores of Bacillus anthracis. The organism reaches the pulmonary bed and causes a necrotizing lesion in the lungs. Furthermore, when the patient coughs out, the organism again spread in the air. Hence, it is transmitted by droplet infection. So it can spread in the community very rapidly. The other form is when this is ingested. It causes gastrointestinal infection, often manifested with bleeding in the intestine and leading to bleeding through pararectal bleeding, nausea, vomiting, abdominal pain, perforation. This could be the presentation. Now, one of the very important features of Bacillus anthracis is that it is an agent of bioterrorism. Why? Terrorism. Now, what is the importance of terrorism? Terrorism is to spread panic among the people. Now, why? Then what will be bioterrorism? When the same act is committed with the agent of which can cause human illness, it is called as bioterrorism. For example, if a chemical gas is released, we call it as chemical terrorism. Okay. So if a biological agent which can cause human disease with significant mortality or morbidity, we call it as bioterrorism. Why it is a very good agent? Because you can make spores of Bacillus anthracis and you can spread in the air. It also causes an airborne disease by droplet infection, so it can spread to the air. Hence, it is a very good agent of bioterrorism. Okay. So, how do we diagnose them? Well, be it a cutaneous lesion or a pulmonary lesion, we take the sample and then we proceed for the microscopic examination on which we can see bamboo like appearance. If you look at this, we can observe this kind of appearance on microscope. Furthermore, what we can do is we can go for cultures on which we can see where various descriptions have been given like medusa head, beaten egg whites, or ground glass appearance, string or pulp appearance. These are seen there. Okay. Now, one very important difference that you should remember is when we should what we do if we want to if we suspect a patient could probably be infected with Bacillus anthracis. That's it. If we suspect a patient is having anthrax, what we need to do? First of all, we need to inform the laboratory that this is a suspected case of anthrax because proper biosafety precaution is needed. Just routinely, if you take the sample and send it to the lab, there is a high, big potential of spreading the disease even in the healthcare workers. So you should be aware of that. Inform the laboratory beforehand before collecting the specimen. And furthermore, while collecting the specimen, you should be aware to protect yourself and your surroundings. That is very, very important. Okay. Just because you think it could possibly be anthrax, you're taking the sample, be it a sputum sample or a cutaneous sample, don't send it to the lab straight away. Consider all other provisional diagnosis, then inform the laboratory, inform the health hospital, uh, whoever the in charge in the hospital is, and then proceed for diagnosis. Okay, so please keep that in mind. In the laboratory conditions, what are the important differences between Bacillus anthrax and other species? Why this is important? Because suppose on culture we do get Bacillus species. Now these Bacillus species are present everywhere in the environment. It could be a laboratory contaminant, it could be a hospital contaminant, it could be present everywhere. But all are not anthrax bacilli. What we need to do is we need to suspect whether it could probably be an anthrax bacilli or not. So what are the indications? The indications are these differences. That is, it is commonly seen that Bacillus anthrax will have a capsule, while other bacilli would not have. Secondly, on blood agar, most of the bacilli will give a hemolytic colony. I think you have understood by hemolytic colony, but Bacillus anthracis do not give hemolysis on blood agar. Furthermore, this medusa head type of colony can be seen with Bacillus anthracis. Now, why this is important? Because in laboratory conditions, if we do find a bacillus on microscopy, and we have seen the capsule and there is no hemolysis on blood agar, that means this could possibly be Bacillus anthracis. When we do arrive to this, we have to send this bacterial strain for further identification in a referral lab with a proper biosafety preparation, with a proper transportation of the specimen and the clinic and the bacterial strain. If any sort of biosafety protocol is broken, it may lead to an outbreak of pulmonary anthrax. Okay, so this is why this is very, very important. So whenever if you do suspect that a patient might be suffering from anthrax, be it pulmonary, be it GI, be it cutaneous, always be aware. Now next comes another bacillus species which is very commonly causing what is known as food poisoning. It could be a preformed toxin or it could be a toxin produced in the gut. So there are two types of toxin. One is the stable toxin and one is the heat labile toxin. The stable toxin, what do you mean by stable toxin? It's a heat stable toxin. So what happens is commonly this bacilli commonly is seen with rice based food preparations. So commonly you have seen fried rice being prepared with a already pre-cooked rice, and then that pre-cooked rice is then stir fried to make fried rice. Okay. Now this pre-cooked rice may be contaminated with Bacillus cereus and it will have toxin. There will be two types of toxins. One is heat stable, other will be the heat labile. Now when we have started preparing say fried rice, that means we are heating at a high temperature. The heat labile toxin will be destroyed, but the heat stable toxin will still be present. Now when we eat that fried rice, what will happen? That heat stable toxin will produce its effect, and almost immediately after eating about 5 to 6 hours, the patient will, what will happen? There will be nausea and vomiting because the toxin is already being present. So this is called as the emetic type. Emetic means which causes emesis or vomiting. Other way around, now suppose without preparing the fried rice, I have eaten the pre-cooked rice. Now this pre-cooked rice will have both the heat stable and the heat labile. Then what will happen? Initially I will have the same kind of presentation, that is the emetic type. After 6 to 8 hours, now I will have the different presentation, that is called as the diarrheal type. Why? Because the heat labile toxin is also called as enterotoxin. That means which when it enters into the intestine, it will start producing inflammation of the intestine and thereby resulting in diarrhea. So Bacillus cereus poisoning can present in two forms, either vomiting or diarrhea or both. So this is a very commonly encountered multiple choice question that you will see in your examinations when you will go for the PG entrance examinations. Very commonly you will be asked about a kind of an emetic poisoning. Most probable cause is the Bacillus cereus and most probably caused by the heat stable toxin. Now let us take another scenario. Suppose a group of children from a school went to a picnic and they had shared a meal. After that, a large number of students started vomiting. So this would be a typical case of Bacillus cereus food poisoning. Now, if you are asked to give diagnosis, how will you confirm the diagnosis? How will you confirm? প্রশ্নটা বোঝা গেছে না বোঝা যায়নি? কি বললাম? Bacillus cereus poisoning present with food poisoning. Suppose 20 school students went for a picnic. They had shared a meal. After that, about 4 to 5 hours, most of the students started vomiting. So what will be the clinical diagnosis? It is a case of food poisoning. Now if you are asked to confirm that this is actually food poisoning, how will you approach? Approach কিভাবে করবে? ভাবো তোমরা এখানে যতজন আছো সবাই দুপুরবেলায় ক্যান্টিনে খেয়েছো, 4 ঘন্টা পরে অর্ধেকের ভমিটিং স্টার্ট হয়ে গেল। এবার আমি জিজ্ঞেস করলাম একজনকে কি করে কনফার্ম করবে যে সত্যিই ফুড পয়জনিং হয়েছে কিনা। হ্যাঁ, মেটেরিয়াল কিভাবে কি মেটেরিয়াল নেবে, কিভাবে প্রসিড করবে? আরে বলো, ঘাবড়াচ্ছ কেন, এটা তো ক্লাস। ওকে, তাহলে ফার্স্টে আমাদের দেখতে হবে হোয়াট ইজ দ্য সোর্স। দ্য সোর্স ইজ দ্য ফুড, so any remaining food we have to test. Furthermore, because presentation is like that of emetic type, that means both emetic and diarrheal type is caused by toxins. Is there any point of doing culture over here? খাবারে কি ব্যাকটেরিয়া ছিল না টক্সিন ছিল? Exactly. So is there any point of doing culture? So what do we need to look? We need to look for toxins in vomitus and the food. Okay. When will be the role of culture? হ্যাঁ, মানে খাবারে ব্যাকটেরিয়াটা থাকতে পারে, যে খাবারটা খাওয়া হয়েছিল তাতে ব্যাকটেরিয়া থাকলেও থাকতে পারে, সেইটা তুমি কালচার করো। ভমিটাস কালচার করলে কিছু পাবে? ভমিটাসে তাহলে কি থাকবে? তাহলে আমাদের কি টেস্ট করতে হবে? উই হ্যাভ টু লুক ফর টক্সিন, দ্যাট ইজ দ্য মোস্ট ইম্পর্টেন্ট। তাহলে in the food we can demonstrate large number of bacilli, but that does not give any kind of confirmation. The best method of confirmation is to show the presence of toxin in both food and in the vomitus. Understood? Okay. So this is Bacillus cereus, how it appears under microscope, slightly shorter in length, and I think you can see some of the spores that are present over there. And on blood agar you can see hemolytic colonies. Can you appreciate hemolysis? This is a blood agar. Can you see the hemolysis around the colonies? These are the hemolytic colonies that are seen very commonly. Okay. These are the typical differences between the emetic form and the diarrheal form. Look over here. The emetic form is intoxication, that is ingestion of the preformed toxin, whereas the diarrheal form is foodborne infection. In the emetic form, probably the best example is rice. The second typical example is a fried rice, when pre-cooked rice is again fried up. So when the fried rice is eaten, if you try to do culture, most of the time you will not get anything. However, toxins will be present. The diarrheal forms are very commonly seen with meat and vegetables, especially in canned meat and canned food items. So mean incubation period for the emetic type will be less than 6 hours, and for the diarrheal type the presentation will be somewhere between about 8 to 12 hours. The emetic time predominant symptoms will be abdominal pain, cramping and vomiting, and in case of diarrheal form the same with diarrhea. Vomiting may also be there. It is not necessarily that in the diarrheal form there will be no vomiting. Vomiting may be there. Okay. And most important difference is emetic form is caused by the heat stable one, and the diarrheal form is caused by the heat labile one. So that was the two most important diseases of the Bacillus species. One is Bacillus anthracis, the other is Bacillus cereus. Now let us move on to the next important is the Clostridia. Now among Clostridia, these are gram positive rods, and they are spore forming, and most importantly they are all anaerobic. Now when I am telling you that a particular organism is anaerobic, that means where does anaerobic organism reside in the environment? কোথায় থাকে অ্যানারোবিক অর্গানিজমস গুলো? নর্মালি কোথায় থাকে? বললাম তো একটু আগে, subsoil, deeper layers of the soil, not in the topsoil. Spores may be present in the topsoil. Vegetative form will be seen only in subsoil and deep soil. Our next important part is intestine of human being and animals. The intestinal area is not aerobic, hence it can survive over there. Now, if you can understand this part, you will understand how clostridial infections occur. All are spore forming. There are various types of spores. One is oval and central spore. Another type can be seen is called subterminal spore. What is subterminal? Just beyond the end, not right at the end, just near the end. This is called as subterminal. This is seen with Clostridium perfringens, which is known to cause gas gangrene. Another type we can see over here is called terminal spore, almost like a tennis racket, right at the end. This is commonly seen with Clostridium tetani. Another form can be seen with oval end. This is spherical, this is oval. So depending on the shape of the spore and the position of the spore, we can make a probable diagnosis. This is very important in case of clinical samples. For example, I think you have read about gas gangrene. What is the typical presentation of gas gangrene? We'll commonly see a person will coming with a history of injury or a road traffic accident in which there is a gross destruction of tissue in the limbs, after which there will be pain, swelling, and a very important part is crepitus. That means formation of gas in the wound. That's why it's called as gas gangrene. Now, to confirm the diagnosis, what we do, we take the aspirated sample and on microscopy, if we do observe gram-positive bacilli with spore, that is subterminal spore, we can give a presumptive diagnosis of Clostridium perfringens. Hence, clinically it could be confirmed as gas gangrene. This is the importance of observing the spores and the morphology. Okay, the most important organisms are Clostridium perfringens causing gas gangrene, Clostridium botulinum causing botulism, Clostridium tetani causing tetanus, and another important is C. difficile. Earlier it was a part of Clostridium, now it is being renamed as Clostridioides difficile. Now let me explain what are the important gas gangrene caused by Clostridium perfringens and other like Clostridium septicus. How does this infection occurs? Any kind of wound injury, be a road traffic accident or a direct inoculation of soil in the wound. Like these injuries can be encountered especially in the people working in the fields, agriculture, or even soldiers who are fighting in the open area. When the wounds are contaminated with the soil, the spores of the Clostridium bacilli will enter into the wound and will start producing the toxin. This toxin will start producing local tissue destruction, which will compromise the localized blood supply. When the blood supply is compromised, it will create an anaerobic environment in which the organism will multiply further leading to toxin production and causing toxemia in the end. This is gas gangrene. A similar style of infection can be seen with Clostridium tetanus, where the organism gets inoculated in an open wound over which it will germinate, start producing toxin, this toxin will be absorbed into the bloodstream. The toxin or the tetanus toxin will then, the very in a simple terms, the tetanus toxin has two parts: tetanospasmin and tetanolysin. Spasmin means it will cause spasm of muscles. So this toxin will attack the neuromuscular junction and it will lead to triggering of the excitatory neurons. When the excitatory neurons are triggered, it will cause spasm of the muscles. Mainly, it will start with spasm of the appendicular muscles. Appendicular means muscles of the limbs. And as the toxemia or the levels of toxin increase, it will then affect the appendicular musculature. Appendicular means the muscles of the body, the paraspinal muscles. When there is severe spasm of these muscles, the patients almost like convulsion with a in spasmodic repeated spasm of contraction of the whole body. Body sometimes might bend in the dorsoventral fashion. It is also called as Dhanush Tonkar in Bengal. So Dhanush Tonkar, Dhanush means like a bow. The body might bend because of the severe spasm of the back muscles. When this kind of spasm happens, what will happen? The patient will not be able to, will be under severe pain, and if it does affect the respiratory muscles, the patient will die of asphyxia. Tetanus may be of many types based on the type of the patients which are affected. It could be adult tetanus, which is commonly encountered with injuries, any sort of injury. Another type was commonly seen is called as the neonatal tetanus. The infection was generally acquired when during giving birth, the neonate or the child acquires the infection from the labor room or the stool of the mother, and the child will be affected with neonatal tetanus. This was called as the neonatal tetanus. The next type of disease is called as botulism. Now, botulism is caused by botulinum toxin. Clostridium botulinum produces this toxin. And this toxin, what does it cause? It causes paralysis. So, if the toxin is absorbed, what will happen? It will cause paralysis of the limbs. If it affects the appendicular muscles, what will happen? There will be paralysis of the legs and the limbs. And if there is severe toxemia, it can lead to paralysis of the respiratory muscles and the patient may die out of asphyxia. A medical benefit of botulinum toxin is also used, which are commonly used for the cosmetic benefit, in which it is given as localized injections for treatment of various diseases, including, say, localized in physiology. You have learned about the tonic-clonic contractions. In many types of neuromuscular diseases, there might be tonic-clonic muscle spasms. Those could be relieved by botulinum toxin. And far more commonly used nowadays is to prevent formation of wrinkles as part of the cosmetic surgery. And finally, coming to the last important disease, it was earlier called as Clostridium difficile, but now it is renamed as Clostridioides difficile. What does it do? This organism is present in the intestine of human beings. So normally what happens is that, are we devoid of this organism? No. But we are not suffering from the disease. Why? Because we have a huge gut flora in our body. So this organism cannot produce the toxin normally. But suppose a patient who is admitted in the ICU and is not taking food, his gut flora will change. Furthermore, this patient is on, say, broad spectrum antibiotics for a prolonged period of time. These antibiotics will also kill the organism present in the gut slowly and steadily. So the gut flora changes, and when this change happens, this organism will proliferate and start producing Clostridium difficile toxin. This toxin will produce diarrhea. So next time when you visit the ICU, try to observe the patients who are admitted for more than 7 days and on broad spectrum antibiotics. Commonly, you will see that those patients will have diarrhea, almost watery diarrhea. That is caused by Clostridium difficile. Now, why this is so important? Because this is a spore forming bacilli, remember? So, whenever the patient defecates the watery diarrhea, along with the diarrhea, will have spores of Clostridium difficile. Now, these spores are very resilient. You need to have high level of disinfectant use in that area. Otherwise, what will happen? This spore will infect the next patient and then will lead to an outbreak of diarrheal illness in the ICUs, which are very, very difficult to control. When we'll be talking about healthcare associated infections, we'll talk about Clostridium difficile infection more in detail. Today, I am just giving you an idea of what it is and why it is so important. Okay, so this is a microscopic finding. This is a direct microscope. You can see pus cells over here and gram-positive bacilli. And if you look carefully, there is a spore. Can you see? And this spore is a terminal spore. So right from this direct microscopic, we can probably say that this is caused by Clostridium tetani. Similarly, on culture and on gram strain, we can see terminal spores, gram-positive bacilli. Okay, furthermore, Clostridium difficile diagnosis is now commonly done with detection of toxin or PCR. I'm not going into the details of that, we'll talk it about later. Now, these were the two most important spore forming gram positive bacilli. Now, let us talk about one or two important non spore forming. The first is, can you tell what is this? এটা কি এটা কি এটা কি বলতে পারবে কি করে বুঝলে কেন মনে হচ্ছে that's a very good point. Chinese letter pattern, and we can see first picture A, staining done only with methylene blue or polychromatic methylene blue. Normally, what do we expect? Uniform staining, right? Gram positive bacilli, they will be stained uniformly. But if we look over here, are they uniform? Some parts are deeply stained, some are not. Because that means there is something which is taking up stain more than the other part of the bacilli. These are the granules or polychromatic granules. On gram stain, you can see there is irregular staining, and the pattern is, like he said, Chinese letter pattern. That is the Albert stain, and on Albert stain we can clearly differentiate the granules from the bacilli. The bacilli will have a greenish tint, whereas the granules will have a brownish tinge to it. Now, what does this organism cause? Commonly, Clostridium diphtheria. It causes, what is diphtheria? First of all, diphtheria is a toxemia. Disease is caused by the toxin. The lesion produced may be orofacial. Orofacial. That is the where tonsils are affected. Other presentation could be What is this? এটা কি? It's an ulcer. আলসার তো কোথায় আছে এটা আলসারটা? হাত পা যাই হোক, মানে স্কিনে। It's a cutaneous ulcer. So it could be presentation with orofacial or cutaneous ulcer. Like orofacial, where the tonsillar bed is affected, where there is a white patch. এবার আমরা ডায়াগনোসিস কিভাবে করি? ডিপথিরিয়া তো পড়ানো হয়েছে, তো আমি পড়িছি, এটা তো বললে হবে না পড়ানো হয়নি। কিভাবে ডায়াগনোসিস করি? ভাবো একটা বাচ্চা এসছে, গলায় ব্যথা, সিমটম দিয়েছে, তুমি মুখ খুলতে বললে দেখলে এত বড় প্যাচ, তার সঙ্গে এখানে কি হয়েছে, এটা কিসের সোয়েলিং? এটা লিম্ফ, মানে লিম্ফ্যাডিনোপ্যাথি। হোয়াট উইল ইউ ডু? থ্রোট সোয়াব নেবে। থ্রোট সোয়াবে কি দেখবে? নেবে তো বুঝলাম, নিয়ে কি করবে? কি টেস্ট করবে? বেশ, গ্রাম স্টেন করবে। আর সেটা বলো, এখানে ছবি দেওয়া হয়েছে, অ্যালবার্সেন বলবে না? এটাই তো আমাদের প্রিজামটিভ ডায়াগনোসিস দেবে। অ্যালবার্সেন আমরা যদি গ্রাম পজিটিভ ব্যাসিলাই উইথ গ্র্যানিউলস পাই, তার মানে কি? প্রবাবলি ডিপথেরি। সেখানে তো আমাদের প্রিজামটিভ ডায়াগনোসিস হয়ে গেল। তারপরে তো কালচার অ্যান্ড আইডেন্টিফিকেশন, সেটা তো পরবর্তীকালে করবো। ট্রিটমেন্ট অফ ডিপথেরিয়া কি হবে? সে দেওয়া যেতে পারে ডক্সিসাইড। আমি অ্যান্টিবায়োটিক জানি কি, কিভাবে অ্যাপ্রোচ করবে, দ্যাট ইজ ইম্পর্টেন্ট। বললাম তো ডিপথেরিয়া ইজ এ টক্সিমিয়া। তাহলে বডিতে যদি টক্সিন থাকে তাহলে আগে কি করতে হবে? সরি, অ্যান্টিটক্সিন দিতে হবে তো। টক্সিন থাকলে অ্যান্টিটক্সিন দিতে হবে না? অ্যান্টিটক্সিনটা কি? ইমিউনোগ্লোবুলিন। আর ব্যাকটেরিয়াটাকে মারার জন্য অ্যান্টিবায়োটিক দিতে হবে। সেটাকে বলা হয় সোর্স কন্ট্রোল। টক্সিনের সোর্সটা কোথায়? এইখানে ব্যাকটেরিয়াটা তো আছে, ব্যাকটেরিয়াটা গ্রো করছে, টক্সিন প্রডিউস করছে, টক্সিনটা মিউকাস মেমব্রেনে অ্যাবজর্ব হচ্ছে। তাহলে টক্সিনটাকে, এফেক্টটাকে বন্ধ করতে হলে আমাকে অ্যান্টিটক্সিন দিতে হবে। আর ব্যাকটেরিয়াটার মারতে গেলে আমাকে অ্যান্টিবায়োটিক দিতে হবে। এই তো ট্রিটমেন্ট। Next, we can go for culture like on slope or potassium telluride agar, and we can also use demonstration of toxin by LX gel precipitation test. ঠিক আছে? Another important part is diphtheroids. Commonly most of the clinical specimen আমরা এই রকম ব্যাসিলাই দেখতে পাই without granules. Those are not diphtheria. Those are other corynea bacteria. Hence, they are also called as diphtheroids. That means like diphtheria bacilli. They are commonly commensals. Hence, they are very commonly contaminants of the clinical specimen or colonizer of the wounds. Wound colonizer হতে পারে। তাদের অ্যাপিয়ারেন্সটা যদি দেখো, এগুলোকে বলা হয় প্যালিসেডিং প্যাটার্ন। প্যালিসেড মানে ব্রিটেনের বাড়িগুলো যেগুলো টিপিক্যাল বাড়ি দেখেছো, বাড়ির সামনে এরকম বেড়া দেওয়া থাকে, লম্বা লম্বা এরকম দেওয়া থাকে, দেখেছো তো? এগুলোকে বলা হয় প্যালিসেড। তো ভালো করে যদি দেখো, এই গ্রুপগুলো পুরো এরকম গ্রুপে গ্রুপে মনে হচ্ছে, এগুলোকে বলা হয় প্যালিসেডিং প্যাটার্ন। দিস ইজ কমনলি সিন উইথ ডিপথেরয়েডস। ডিপথেরয়েডস ডু নট কজ ডিপথেরিয়া। ভুল করে যদি পরীক্ষার সময় এটা না বেরো। আচ্ছা, নেক্সট আরেকটা কি বললাম? অ্যাক্টিনোমাইসিস। They cause wound infections, commonly seen with infections of the jaw and the mandible, and sometimes intestinal infections. On microscopy, they appear like long filamentous structures. These are not fungi, they are bacteria. Another filamentous bacteria is called as nocardia. This is also a gram-positive bacilli. Furthermore, this is also acid-fast. However, the percentage of acid used is less, which is 10 by modified staining. This causes nocardiosis. The most common presentation of nocardiosis is pulmonary nocardiosis. Okay? Lung infection caused by nocardia, commonly seen in immune compromised patients. এর বেশি আমি এখন যাচ্ছি না। আচ্ছা, এখানে দেখো, এটা একটা ক্লিনিক্যাল স্পেসিমেন, wound swab নেওয়া হয়েছিল। wound swab-এ যদি দেখতে পারো, এখানে হচ্ছে কি টাইপ অফ সেলস দিস আর এই হিস্টোলজি পড়ে পাস করে এসছো, প্যাথোলজি পড়ে কি টাইপ অফ সেল বলতে পারছো না? এগুলো স্কোয়ামাস এপিথেলিয়াল সেল। এবার এপিথেলিয়াল সেলের কাছে এখানে দেখতে পাচ্ছ গ্রাম পজিটিভ ব্যাসিলাই। They are uniformly stained and are arranged in haphazard pattern. So these are the commonly how diphtheroids are seen. ঠিক আছে? They are very common because they are also part of the skin commensals. ঠিক আছে? Now this is another type of ডাইরেক্ট কালচার থেকে গ্রাম স্টেন করা হয়েছে। এখানে যদি দেখো গ্রাম পজিটিভ ব্যাসিলাই ইউনিফর্মলি স্টেন দেখতে পাবে। সিমিলারলি এরকম ব্যাসিলাই আরেকটা যেটা হতে পারে, যেটা বারবার বলছিলাম, সেটা হচ্ছে there are many types of same bacilli অ্যাসিড ফাস্ট এনে এটা কি রিয়াকশন দিয়েছে, এরকম দেখতে পিঙ্ক কালার, that means these are acid fast. So there could be many bacilli যেটা গ্রাম পজিটিভ, it could also be mycobacteria. Mycobacteria মানেই যে Mycobacterium tuberculosis সেটা নয়. There are many types of other mycobacteria which cause human disease like non-tuberculous mycobacteria. Commonly pulmonary infection করতে পারে বা skin infection করতে পারে। ঠিক আছে? So this is the overall type of gram positive bacilli. ঠিক আছে? So to recapitulate, আমরা কি বলতে পারি, gram positive bacilli can be either spore forming or non-spore forming. Among the spore forming, the important are the bacillus and the clostridium. The bacillus is aerobic and the clostridium is anaerobic. Among the non-spore forming, the important would be the one which are acid fast. Among the acid fast are the mycobacterial species. Non-acid fast would be Listeria, Erysipelathrix, and irregular bacteria. Among the irregular, we have Actinomyces and Nocardia. ঠিক আছে? So this is the all gram-positive bacilli in a nutshell. Now you will all move to the practicals and do the practical like last class.